So, welcome to my short poster, which is about the infestation of commercially rare bay scallops by the shell-boring polychaete Polydora neocaeca on Nantucket Island. So, shell-boring polychaetes are very interesting in that they are a group of polychaetes that are capable of burrowing into the calcium carbonate shells of mollusks. And they are known to be a pest of commercially reared mollusks such as oysters, abalone, and scallops. So what happens is that these worms burrow into the shell, into the nacreous layer of the shell, and then the animal must now divert energy from important processes such as growth and reproduction into shell repair. And so these pictures gives you an idea of some different species that are known to burrow into these shells. Polydora websteri is probably the most notorious of all of these shell borers. It's implicated in more than 95% of shell infestation cases worldwide. So this study began because of a connection that I had with shellfish farmers and scientists on Nantucket Island. So for those of you who do not know, Nantucket Island is an island located off the coast of the New England region of the United States. So it's in the Atlantic Ocean. It belongs to the Commonwealth of Massachusetts. And one of the important activities that occurs on the island is scallop farming. In fact, Nantucket actually has the last remaining wild-based scallop fishery in the world. Not only is it economically important to the residents of Nantucket and to the Commonwealth of Massachusetts, but it also has very strong cultural importance. Generations upon generations of the island residents have been involved in scallop farming. It's one of the ways in which the residents can supplement their income, and Native Americans have also scalloped long before that. Now, this study began because there have been researchers at the island and farmers who have reported increased incidences of mud blisters on their scallops. Whether or not this is because there's a new introduced shell-boring polychaete, or whether it might have been a polychaete that is increasing its infestation rates because it's tied into some environmental factor like warmer water temperatures, we don't fully know. And so what we decided to do was try to figure out what's actually going on. And so we wanted to quantify parasite load in the bay scallops, and we also wanted to determine the identity of the polychaetes responsible for shell infestation. We used DNA barcoding because many of these shell-boring polychaetes belong to species complexes. And so it's very difficult to identify them morphologically. And so for some complexes, genetic barcoding is the only alternative. And so we collected about 30 scallops from the Brant Point hatchery, which is actually farmed scallops and oysters. So we got them from these cages that are submerged from the docks attached to the hatchery. And here you can see what an infected and an uninfected scallops look like. The red arrows point to mud blisters. Right? And so we broke the shell and we were careful to slowly extract the worm. So in this picture over here, you can see the worm that is snugly fit into the burrow. We identified it first morphologically using polychaete keys, and then we extracted DNA from about 8 individuals. We amplified the cytochrome C oxidase 1 gene, and then we carried out some preliminary phylogenetic and population genetic analysis. So for our results, we found that the prevalence was about 40%, and the mean intensity was about 3.83 individuals per infected scallop. So overall, the parasite load was relatively low compared to what's been reported from other places, not only in North America, but around the world. So our genetic analysis, we did an initial genetic comparison with the GenBank database using the BLASTing tool, and we got about 99 to 100% similarity with our specimens to a species called Polydora neocaeca from Rhode Island. Now, this was interesting because Rhode Island is actually the type locality for Polydora neocaeca, and this species has actually never been recorded from wild bee scallops before. We built a phylogenetic tree, and we found that our 8 individuals actually formed a nice clade, well-supported clade with other individuals from the eastern United States and from North America in general. In fact, we confirmed reciprocal monophyly with specimens from Rhode Island, which is the type locality. We also included in our analysis individuals from South Africa, which formed their own clade, and also individuals from Japan. So this is called Polydora haswelli, but haswelli has now been synonymized to neocaeca in a previous study. Now, the next thing we wanted to do was we wanted to figure out the genetic interrelationship. So dig a little bit deeper to figure out the levels of gene flow between populations. And so our haplotype network gives us an indirect estimate of connectivity. So each of these circles represent a haplotype. And you can see that we have a large haploclade that sort of corresponds to our phylogenetic clade, which we expected. What was interesting is we had a single haplotype that was shared by individuals from Rhode Island and from Nantucket, meaning that there was some level of genetic exchange occurring between the two populations. Now, if you go back to the map over here, you notice Rhode Island is located on the mainland of New England. Now, because of the oceanographic conditions in the area and because of the type of larvae that Polydora neocaeca produces, it's very unlikely that this connection was made naturally. What could have happened is that oysters, infected oysters or some infected mollusk could have arrived in Nantucket carrying Polydora neocaeca, which then could have infected wild bay scallops. We know that Nantucket in the past have imported spat from other farms in New England. So that could be one possible explanation of how the worm got there. However, there's a lot of things that need to be investigated. What I just presented here is unpublished data. It's very new, we only just got it. And so we're looking at future studies in terms of recovering additional populations in New England that we can use in to create a much larger phylogeographic study. And also one of the things we want to do is we want to monitor the prevalence and the intensity across multiple months, preferably across multiple seasons. Because all of the data, the parasite load data, represents a single data point, and that is May 2021. But we want to see what happens as the water temperature begins to increase and decrease throughout the year. And finally, I would like to acknowledge the Nantucket Biodiversity Initiative and the Nantucket Shellfish Association that funded this study, and also the Brand Point Hatchery and the town of Nantucket, which were our connections to the island. And thank you for taking your time to listen to this.